Hi everyone, I'm excited to be part of this meeting. Thank you for the organizers for putting everything together, especially with everything going on right now. My name is Nicole and I'm a PhD student in the Bergstralh lab at the University of Rochester. Today I'm going to tell you about a project that's actually been rapidly evolving in the lab since quarantine due to it being mostly computationally based, and it involves a tool that we've developed in order to analyze epithelial architecture. So just to orient you to my talk to begin with, I'm going to kind of split this into three sections. First, I'm going to briefly go over some background on our lab's interests. Second, I'll go over the tool and how it works. And then lastly, we'll look at what we've learned from the tool so far. So what are we interested in as a lab? Our lab's interested in how epithelial tissues are built and maintained. So as many of you probably know, epithelial tissues are the most common tissue type in the body. They come in different flavors, so to speak. So, for example, we have simple columnar epithelia that line the intestine, and we have simple squamous in the lung. But all epithelial tissues are sheets of cells that line surfaces of the body and provide protection from the outside environment. And oftentimes the sheets of cells are organized into tissues with specific shapes, and these shapes are essential for function. So here are a few examples. Coming back to the intestinal cells, these are organized into both crypts at the bottom here and villi, and this serves two functions. First off, the crypts, which house the stem cells down here, are protected from the lumen. So that's obviously important so that the stem cells can keep replenishing the cells that are shed from the top of the villi. And then the villi themselves protrude into the lumen to increase the surface area and maximize nutrient uptake. Second example here is the kidney epithelia. These are organized into tubules, and the shape of the tubule and the organization vary depending on where in the chain of the kidney they are, but they're all optimized for their specific function. And last off, the alveolar cells in the lung are simple squamous, and this is optimized for gas exchange. So in all of these examples, the organization of the epithelial cells is actually essential for the organs to function properly, and misplaced cells are thought to lead to diseases such as cancer. So how do cells organize into these shapes? There are many cellular behaviors that drive tissue shape change and development, but as a lab, we're particularly interested in these bottom two here, so cell division and cell integration. The cell division in particular poses a problem for a tissue in that the cell undergoing mitosis is greatly changing shape as it's growing and rounding, and then it has to undergo cytokinesis, and the two daughters have to be placed within the tissue, all without compromising the integrity of that tissue. And just to emphasize that point, here are two movies of cells that are undergoing mitosis. The first example here is Drosophila follicular epithelium, while on the right is a mouse intestinal organoid. In both movies, the membranes are marked in green, and then tubulin is marked in red. And you can see that in both, the cells have rounded, moved apically in the tissue, and then the two daughters are born slightly out of the plane of the tissue. Here it's rounding towards the lumen, and then, but in both examples, after division, the two daughters return basally and reincorporate back into the tissue. And all while the rest of the tissue isn't affected, everything remains intact and everything's still functioning well. So we are using MDCK cells that form monolayers to look at how epithelial layers are built. So MDCK cells are derived from a dog kidney, and as you can see from this fixed imaging and from the movie, which I'll play, here we go, the sheets of the cells undergo many divisions even when the tissue already looks pretty packed. So this looks pretty regular, but you can see here highlighted many, many cell divisions. And this is all happening without these other cells being compromised. Just to look at the side view of these, when we grow MDCK cells on a collagen-coated slide, they form polarized monolayers in culture. So they're not inherently polarized, but we can induce polarity. And here you can see that there's a nicely packed sheet of cells, and if we look at the XC view, we can see that ABQC is localized apically, so a marker of polarity, apical polarity here. And also then just to point out, we use phalloidin to stain for actin, and then DAPI for the DNA as well. So these, monolayers look quite good, and by looking at them we could tell they're a layer. But actually we see a lot of variation in the organization across the slide. So We can see areas that are very sparse and very flat. We see nice areas like the example I just showed you, and we can also see areas where it looks quite disorganized. So this implies that the development of the layer isn't uniform across the slide. And so we need a way to classify these layers that is in an unbiased and consistent manner. And that's where our goal comes in. So this, the whole point of this project is to be able to characterize MDCK monolayer development in an accurate and unbiased manner. And that brings me to part 2 of my talk, which is what is our tool and how does it work? So just to quickly go over what we expect an MDCK monolayer to look like, I've shown you a few examples, but just to go into it in a bit more detail. When the layer is formed, we see this nicely packed, organized cells. And then if we look at the XZ view, we can see that the nuclei are all towards the bottom of the tissue, and the actin is enriched along the lateral and apical surfaces. Sorry. So we know this by just looking at the layer, but we need a way to computationally import this information pixel by pixel and determine where the layer is in the image. So to do that, we created a pipeline for image acquisition. So first off, we plate our cells on a collagen-coated slide, as I've said, and then after 24 hours, we fix and stain the layers. And then we image the same 16 positions within the well. So this, as I said, the organization across the well doesn't appear to be uniform. So we don't want to be picking and choosing where we image. And then once we have these images, we use the cells tool in Amaris to segment out all of the nuclei in the image, shown down here. And this then gives us the X, Y, and Z positions of all of the nuclei as well as the volumes. So then we can use this information as well as the original corresponding image file to load into our automated layer analysis tool, which is Alan. So the first step on here is to go through the nuclei and just filter out anything that has been segmented that isn't a true nucleus, so anything that's probably debris or anything that's just crazy too big, so that we get all of the signal that seems to be real nuclei. And then we run this along with the image file through the rest of the script, and this determines where the layer is by looking at both the positional information of the nuclei as well as the actin signal. So we go through step by step in Z, summing the X, Y actin signal, and this gives us the blue profile here. And then we can set the bottom and the top cutoffs based on that actin signal. So I'm not going to go into any more detail specifically just for time's sake about how this works, but if you have any questions, feel free to email me. And we went through just to make sure that this seems to be working well, and a few of us in the lab took some examples, said where we thought the top and the bottom of the layers were, and compared that to Alan, and we matched up pretty well. One thing I just want to point out here as well is that there is quite a lot of variation in our manual determination of the top and the bottom of the layer, so this just emphasizes the fact as to why we want to actually do this computationally. But overall, we matched up pretty well with these measurements that the tool is giving us. So we can also tell from these images and from loading everything through the script whether or not our cells have actually formed a layer. So we can go through and then, based on where the nuclei profile is in a relationship to the actin profile, we can say whether or not the layer is actually too flat, so it's not actually a proper layer, it's probably an unpolarized layer. We can say whether it's too disorganized, so this one looks like the cells can just kind of clumped up on top of one another rather than forming a nice layer, and you can see that the actin profile looks pretty crazy on this side. And we can also just, only then focus on the ones that are just right for the analysis that we wish to do. So just to point out here, this works really well on examples that form either really flat layers, very disorganized layers, or are very clearly a layer. The borderline examples are giving us a little bit of trouble right now, but we're optimizing that. So I just want to point that out, and then so for that reason, the rest of the presentation has also been manually verified, all of the data. So coming to that, what is it that we've learned from Alan so far? So one of the things that we actually can get from Alan is that we can distinguish between developing and mature layers. So we see that at 8 hours post-plating, the image is classed as a layer according to Alan. and all of the nuclei are towards the bottom of the actin signal, and we get the nice actin curve here. But as we go through time, we start to see this little shoulder appearing in the actin signal and a sharper peak for the apical signal here. So we can see that in the projection as well, that the apical signal seems to be becoming more intense, as well as the lateral signal, as the layer becomes more organized. So as it becomes more regular, we get to see this specific profile in the actin, which is pretty cool. So another thing that we have found from Alan is that between 8 hours and 24 hours, when we plate 200,000 cells, we actually see that at 8 hours, none of these images are classed as a polarized layer. The majority of them are actually unpolarized and flat. But over 24 hours, we see a good proportion of these actually becoming polarized layers. So this is good that we can actually work this out, but we wanted to know whether this polarization is actually due more to cell proliferation or reorganization of cells over time. So in order to answer this question, we need to tease apart time and density and layer development. So to do this, we can do various different manipulations to our cells. So we can change the initial plating number, either 100,000 versus 200,000. We can change the time, 8 hours, 24 hours, or 48 hours, or we can actually change and stop cell division. So we can use a G1 inhibitor to stop the cells from dividing, to then see just what contribution the reorganization of cells can have over time. So we did these 3 different manipulations, but for each one, the data I'm going to show you is all the same final density. So this way we've decoupled density from time or cell number, initial cell number that is, and division. So we're looking at all of the same final density, but we've got there in different ways. So when we look at that data, we actually see that if we treat the cells with riscovatine, which is that G1 inhibitor that I just was saying about, we see that actually none of those over 24 hours form layers. So all of them are unpolarized or disorganized. So, but if we look at the same plating number initially and the same time period, we see that actually that a lot of these form nice polarized layers. So this way we've uncoupled time, cell number, and just looking at cell division here, and it seems that cell division might actually primarily be driving the organization of the layer. And it's not really the initial cell number or time that matters. So that's kind of cool. We want to look into this in a little bit more detail, and how we're going to do this is to build a computational model to further tease apart the relationship between division time and layer development. So by doing it this way, we can manipulate individual variables and look and see what combination of variables actually are giving us the layers that we see. So with that, I'll just conclude. So we have developed a custom script that determines whether or not MDCK cells are formed a nice monolayer. We can determine how many nuclei are within that layer and on top of the monolayer too. I didn't go into that with so much detail, but we're doing a lot of work with that. And we can look and determine the stage of the layer development. So the number of extralayer nuclei also increases with cell density. I didn't show you this, but we are looking very much into whether or not the extralayer nuclei are there because of cells just landing on top initially, and cells landing on top and then dividing, or cells leaving the layer, or both. So we're looking at that. And we also have seen that organization is actually independent of cell density and time, but definitely requires cell division. So moving on, we want to use this computation modeling to further investigate this relationship between cell division, time, and layer formation. We're currently making our script more user-friendly and doing that optimization that I mentioned at the beginning, so it can be more accessible to other people to use. And we are starting to look at our lab's molecules of interest, primarily cell adhesion molecules, and we can manipulate these individually and then see how they also affect layer formation. So with that, I'd like to thank my lab. This is how we've all seen each other recently during quarantine. In particular, I'd like to thank another PhD student in the lab, Christian, who's helped me a lot with the coding. I'm not a computational biologist, so without him, this would have been a lot slower. And with that, if you have any questions, feel free to email me. Thank you for listening.